Ladies and gentlemen, Nobel Prize in Physiology or Medicine. I will first read the announcement in Swedish, followed by English, and after that we will have some time for presentation of some background and, of course, to open up for questions. So I'll start in Swedish. Nobelförsamlingen vid Karolinska institutet har idag beslutat att Nobelpriset i fysiologi eller medicin år 2019 ska delas lika mellan William Kaelin, Sir Peter Radcliffe och Greg Semenza för deras upptäckter av hur celler känner av och anpassar sig efter syretillgång. Så det var svenska. I will turn to English. The Nobel Assembly at Karolinska Institutet has today decided to award the 2019 Nobel Prize in Physiology or Medicine jointly to William Kaelin, Sir Peter Radcliffe, and Greg Semenza for their discoveries of how cells sense and adapt to oxygen availability. Here we have the three happy laureates. To the left, Greg Semenza. He was born in 1956 in New York. He performed his prize-winning studies at Johns Hopkins University in Baltimore, where he's still active. In the middle, Sir Peter Radcliffe was born in 1954 in Lancashire in the UK. He performed his prize-winning studies at Oxford University, and he's continuing to do his research at Oxford University, and he's also at the Francis Crick Institute in London. And to the right, William Kaelin, born in 1957 in New York. He performed his prize-winning studies at the Dana-Farber Cancer Institute in Boston, where he's still active in his own lab. I will now turn to Professor Randall Johnson, a member of the Nobel Assembly, who will describe some detail and background behind the work. So please, Randall.
This year's Nobel Prize is awarded for determining how oxygen levels are sensed by cells. Oxygen is essential for life and is used by virtually all animal cells in order to convert food to usable energy. However, the amount of oxygen available to cells, tissues, and animals themselves can vary greatly. This prize is for three physician scientists who found the molecular switch that regulates how our cells adapt when oxygen levels drop. The most fundamental use of oxygen by the cell is to convert food to usable energy. Just as a candle needs the right amount of oxygen to burn cleanly, cells need to adjust their metabolic rates based on how much oxygen they have available to them. This allows each cell, and indeed our bodies, to efficiently and safely burn fuel so as to create heat, do work, and build new tissues. Cells and tissues are constantly experiencing changes in oxygen availability. As an embryo grows and develops, as muscles work, the oxygen available changes as the tissues themselves change. Cells need a way to adjust to the amount of oxygen they have while still doing their important jobs. Sometimes, oxygen levels change across the entire body, such as when we go to high altitudes, and sometimes they change in very small parts of our bodies, such as when we get a wound that interrupts the local blood supply. This triggers an adaptive process called the hypoxic response, which in turn can induce processes in the body as diverse as new blood vessel formation, or angiogenesis, new red blood cell formation, or erythropoiesis, and metabolic adaptations of cells, including glycolysis. One of the first physiological changes caused by low oxygen noted by scientists was the erythropoietic response. This occurs when the body is at high altitude or when it experiences anemia, such as an insufficient number of red blood cells. This hypoxia, or low oxygen state, induces expression of a hormone by the kidney, the erythropoietin hormone, also known as EPO. It became clear to researchers in the field that EPO gene expression goes up when oxygen levels go down. This allowed scientists to use the EPO gene as a tool to analyze the role of oxygen in the control of gene expression. Now we turn to the work that gave rise to this year's Nobel Prize. The earliest work by one of this year's laureates, Greg Semenza, showed that this increase in EPO was due to a specific region of the EPO gene that he defined and called the hypoxia response element, or HRE. Semenza used this oxygen-regulated segment of the gene to isolate a transcription factor protein complex. He called this protein complex HIF, for hypoxia-inducible transcription factor. HIF itself is formed of two proteins, one called ARNT, or ARNT, that is always present, and one he called HIF-1 alpha. The HIF-1 alpha protein is oxygen sensitive, which is to say that it is present when oxygen is low and disappears when oxygen levels are high. Semenza and others showed that HIF was present in almost all cells, and at this point HIF was potentially a key regulator of oxygen responsiveness. Further work by a number of groups showed that HIF was constantly made by cells, but in high oxygen conditions, which we call normoxia, it was constantly destroyed by proteasomal degradation. At the time of the cloning of HIF by Semenza in 1995, William Kaelin was studying a gene involved in cancer called the von Hippel-Lindau gene, or VHL. His group, in collaboration with others, showed that cells lacking VHL had increased expression of hypoxia-inducible genes. Subsequently, Sir Peter Radcliffe's group showed that VHL was necessary for HIF to be destroyed by the proteasome. This indicated that a key role for the VHL gene was to direct HIF to be destroyed under high oxygen conditions. However, we still didn't know how oxygen regulated this switch. Radcliffe and Kaelin then simultaneously showed the way by demonstrating that an oxygen-dependent reaction seals HIF's fate in high oxygen conditions. When enough oxygen is around, two prolines in the HIF-1 alpha protein are hydroxylated. Hydroxylated HIF is then recognized by the VHL complex, which then targets HIF for destruction. To sum up, then, this elegant switch, which was elucidated by the work of this year's laureates, under low oxygen conditions, HIF is made, it accumulates in the nucleus, and that results in the induction of a wide variety of oxygen-sensitive genes. But when oxygen is present, HIF is hydroxylated, recognized by VHL, and destroyed. There are at least 300 genes with hypoxia response elements, or HREs, in the human genome. These are all regulated by HIF to help cells, tissues, and the entire body cope with changes in oxygen levels. Some of these genes include those encoding the vascular endothelial growth factor, or VEGF, that regulates angiogenesis, erythropoietin, and almost all of the enzymes involved in glycolysis, or anaerobic fermentation. The effects of HIF range across almost every aspect of physiology, from metabolism to exercise, to embryogenesis, to immunity, and the changes wrought by altitude. Diseases where HIF response plays a critical role include anemia, cancer, stroke, infection, and heart attack. Applications of these findings are already beginning to make their way to the clinic, with potential drugs to increase HIF used to treat anemia and to suppress HIF used to treat some forms of cancer. These fundamental findings have greatly increased our understanding of how the body adapts to change, and applications of these findings are already beginning to affect the way medicine is practiced. This year's 3 laureates have greatly expanded our knowledge of how physiological response makes life possible.
Thank you very much, Randall. So we'll turn to questions, and to my help here today, we have the chair of the Nobel Committee, Professor Patrick Ernfors. We have member of the Nobel Committee, Anna Wedell, and we have Randall, member of the Nobel Assembly, an expert in this field as well. So we'll open up for questions, so please go ahead. Here we have one up there.
Hi, I'm Anneli Beckner. Do you want me to speak in Swedish or English?
I think it's appreciated with English, but if you have questions and someone wants specifically to ask in Swedish, that's okay too.
Okay. I'm from Swedish TV4, and I would like to know a little bit about, you said it treats cancer, for instance. Why is it important that this
EPO.
Yeah, the EPO and those proteins, why is it important what they do when it comes to cancer?
Okay.
These proteins,
Okay.
good or bad?
Yeah, no, I think if I'll ask Anna Wedell to answer that question.
I should start by saying that the protein you mentioned, EPO, was just sort of the initial protein that was studied as a tool to find this whole system. And there are hundreds of genes that are regulated by the system. And in cancer, there are many links because cancer cells and tumors can exploit this system to optimize the condition so they can grow rapidly, to put it simply.
One could mention that one thing, a tumor in the body, for example, lacks often blood supply. And if it activates this response, it helps the tumor to attract blood vessels to supply more oxygen and nutrition and so forth. But so it's several aspects of cancer cells that...
So oxygen is used differently when you grow rapidly, for instance. Your metabolism will be different. So they will use the system to direct metabolism to be able to grow and also make blood vessels.
Okay. Any more questions? I'm sure there'll be a few. Please go ahead.
This is David Keaton from the Associated Press. We understand you've spoken to some of the laureates. What was their reaction when they heard that they have won this prize? And what's their, who have you spoken to, and were they somewhat expecting this?
I'm happy to say that I was able to reach all 3 of them, which sometimes is a little bit difficult, but that was successful. I should say that the last one I called was William Kaelin, who we didn't have a number to, so this morning I was able to reach first his sister, who I woke up, and she gave me 2 phone numbers. I called the first one and asked if I was speaking to Bill Kaelin. No, the person said, and it was the wrong number. But the other phone number was correct, so I was able to reach Bill Kaelin. He was really happy, almost speechless, and the other 2, the most awake person, I think, was Sir Peter Radcliffe, who was actually in his office. But also Greg Semenza, I think, was asleep and was extremely happy to hear the news, and happy to actually, they mentioned also they were happy to share the prize with each other in this trio. Some more questions? Sure, go ahead.
Maybe if I...
Yeah.
Ask another one then. So for people that are not well versed in the science that will be hearing this, what will be the most easy to grasp benefit of this discovery to them, to the person listening to the news this morning?
Right. I'll think if Patrick Ehrenfors could respond to that.
Well, I mean, it's a system which is required for our body to function normally. So oxygen levels vary in different parts of the body, in tissues and organs. And in particular, for example, during exercise. The muscle during exercise become anaerobic. So this system both is dual. It both makes the cells cope during low oxygen levels, but it's also a system that generates an opportunity for the cell to regain normal oxygen levels. So it's an adaptation of the cell's metabolism to cope with low oxygen, but also a system that increases blood vessel growth into the tissue and also increases red blood cells that now can transport more oxygen and more efficiently transport oxygen. So it's both of these systems. And of course, this is such a basal physiological mechanism that allows us to colonize the Earth in different parts at different altitudes, because oxygen levels vary at different altitudes. So obviously this system is involved there as well. And when you have this kind of basic, fundamental processes in your body, of course, those are also really important in diseases, as we heard Randall mention, such as, for example, anemia, or cancer, or heart attack, or stroke, or other disorders where you have a reduction in blood supply and oxygen and nutrient supply.
So really short, one could say that this is one of the critical systems or adaptive systems for animal life overall. So it's an incredibly central, important adaptive system for animals. Something else? Here we have one in the middle.
This is Nordic Chinese Times, and thanks for the announcement and the presentation of this year's Nobel Prize in Physiology or Medicine. As we can see, this year's Nobel Prize is related to fundamental research. It is always useful to apply the findings to the application to our humankind, right? So I'm wondering what is the breakthrough of this finding in treating the cancer, and I'm also wondering the possibility of applying this knowledge to develop any early cancer diagnosis or the potential treatment. Thanks.
So let me ask Randall Johnson to respond to that question.
Okay. Oh, I guess I don't need to turn this on. Well, there are fairly immediate applications that are in development at the moment. It's such as Patrick pointed out and as Anna noted, this is such a fundamental aspect of how our cells deal with getting oxygen and then adapting to its use that one could almost imagine the applications will be, I don't want to say endless, but very, very wide. The very first applications that appear to be coming close to clinical use, in fact, one was just approved in China, is the use of increased HIF expression to treat anemia. And the idea there is that as HIF goes up, not only does that increase the level of erythropoietin production, which of course stimulates red blood cells, but HIF also controls iron metabolism. And so we'll really coordinate how the body takes up iron and then how it incorporates that into the growing red blood cells. So these are, of course, important applications both from the standpoint of people with anemia, and oftentimes people with anemia have many other problems, including kidney disease or even cancer, and so it may be an application that's useful there. The other thing you alluded to was the treatment of cancer itself, and interestingly, as Thomas pointed out, and as my colleagues pointed out, tumors have increased HIF expression, likely to help them cope with low levels of oxygen in their interiors. And so there are drugs in development to actually inhibit HIF, to allow tumors to be shrunk, since tumors use HIF to expand. Now, that may be difficult to conceptualize that something could be both bad and good. But of course, that's what a basic discovery is. It shows every aspect of, or it impacts every aspect of the body, and certainly future work in this field will likely have wide-ranging impacts.
Right. Are there any additional questions? I know some are eager to interview, but please go ahead.
Hey, this is Nordic Chinese Times. I'm very curious about the biggest argument or concern when you are reviewing the nominees and the projects.
Okay, maybe I could briefly answer that. It actually relates to Alfred Nobel himself and what he wrote in his will, and what really constitutes the basics of our rules, regulation, and statutes. And that is to give the prize for a discovery in physiology or medicine. So each year we are totally focused on awarding the really most important discovery of all the proposals we have on the table each year. So that's really, really central, and that is what is the sole determining factor, I would say, in our discussions and decision process. Anything else? One more here. Maybe you can take the microphone.
Fred from China's Xinhua News Agency. Just want to confirm very quickly, the prize amount for this year's prize is also 9 million Swedish crown, right?
I didn't get it.
The prize amount this year is also 9 million Swedish crown, right?
The prize
Amount.
it will be 9 million
And
Swedish
equally
kronor.
divided between the three.
And that will be equally divided between the
Thank you.
3 recipients, yes. I think if there's nothing else that we can close this session, and some have already booked interviews, and we are also available for additional interviews for anyone who is interested in that. So thank you so much for coming here today. Thank you. Hey.